indicates the existence of genetic diversity. Crossbreeding process can cause increased allele variation derived from genetic combination of female and male parents. Conclusion: primer jumps 839, jumps 880, and jumps 864 are informative and could be considered as a suitable marker to analyze genetic diversity in Chile F2 population from crossing Laris and SSP. At similarity coefficient of 0.74, there are 5 clusters. Cluster 1 consisted of 29 progenies with Laris. Cluster 2 consisted of 8 progenies and SSP, while 5 progenies were in separate clusters. That's all of my presentation.
Thank you very much, Ms. Yulis Sulistiawati. Next, we move to the next presenter, Ms. Penny Lestari. She will talk about the thermogradient in a rice germplant. So, Ms. Penny Lestari, screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. I deliver my thanks to moderator for the opportunity. The screening rice using thermogradient pair is our work in our previous affiliates, namely LIPI and Ministry of Agriculture. Since this year there is a change of affiliation, so let me introduce our team. I, Penny Lestari, with Dr. Untung Susanto and Indra Gunawan, now joining to BRIN, while Ms. Olga Ratna Roshaini was joining to IAAIS, part of Ministry of Agriculture. This research is motivated by the awareness that in recent years there has been a significant increase in the air temperature. Until NOAA released data that 2021 was a part of the 6 hot years in the last 40 years of data, and this temperature increase mainly occurs in Asia, where rice is mostly produced. There will be decreased rice production. The assembly of high temperature tolerant rice is one of the important strategies for mitigating famine caused by climate change, because rice is eaten by the majority of world population. Recent years, plant breeding for temperature tolerance rice related has been carried out using tree selection facilities, namely open field involving multiple patients. In this condition, the selection and timing cannot be controlled. And the second one is using growth chamber facilities. Of course, these two options is costly. Therefore, we offer our tools called as thermogradient bar. These two work based on temperature gradient. The temperature gradient from 2 to 45 Celsius, and line selection can be carried out simultaneously at various temperature. Okay, so the use of thermogradient bar is a solution to streamline high temperature tolerant rice breeding program by conducting selection at the earlier stage, reducing the number of genotype tests in the field and reducing selection costs. The study aims to determine the cardinal temperature of rice germination, determines a suitable environment for the high temperature tolerance site, and select high temperature tolerant rice lines. To cope the aim, 33 rice lines which were tested from various breeding programs. All the seeds are ICRR collection, we call SBB padi, and tolerant check varieties were N22 and Bio, which were genotype introduced from ICRR and ASEAN a corporation project, while the sensitive check varieties were Ciherang and Huang. Both of them are popularized varieties in Indonesia. We used RCBD experimental design with 5 replication based on time. The number of seeds per replication was 10 seeds, adjusted to the chamber capacity. Then all genotype viability 100% based on tetrazolium test. The seeds were planted on the thermogradient bar plate using 3-layer tall tissue as a medium at a totally dark condition. The environment remains stable during the treatment because the thermogradient bar took place in closed system. Temperature and humidity check were carried out automatically through the thermohygrometer that is plugged into the thermogradient bar column. Observation was conducted for 9 days, including optimum germination temperature, critical temperature for a high temperature stress selection environment, and select the best genotype. All decisions were based on germination and normal seeds. Okay, the seeds is a normal, while the seed produce at least 2 millimeter radical. The number of sprouts was counted every day until 9 days after sowing. We move to the result. The cardinal temperature is defined as the temperature at which at least 50% of the seeds germinate. So we declare from our result that 20 to 40 Celsius is the cardinal temperature for rice germination, while 10 Celsius strongly inhibits germination. We found that 30 Celsius is the optimum temperature. Also with 20 Celsius, we found that highest standard germination from 40 Celsius is the highest than other treatment. So we conclude it was the selection environment for rice line to high temperature tolerance based on percent germination. Okay, from 36 genotypes, and 2 with Gaia Bio are at the top, while HT11, 12, and 18 is the bottom of the bars. HT25 is the most high temperature tolerant genotype of this population, exceed the check, while Ciherang and Huang is the moderate tolerant to high temperature. Based on box analysis, we find that HT6 to HT27 is the tolerant genotype. As said earlier, that the thermogradient bar is very helpful for selecting high temperature tolerance, including in rice, as well as low temperature. Interestingly, in this study, we found that genotypes including moderate temperature, this is HT 31, 34, 32, and 36, is also highly tolerant to low temperature. So those genotypes can be selected for assembly the varieties with a wider distribution. This research also note that 40 Celsius and 10 Celsius did not produce leaf seed until the end of the observation, as well as in the check, so that validation of the tolerance of the selected genotype in the field is still needed. Okay, so our conclusion is thermogradient bar is effective as a selective tool for temperature tolerance on rice, and we do recommend it to other species. Then 20 to 30 Celsius is the optimum germination temperature for rice, while 40 degrees Celsius is suitable as a selection temperature for screening high temperature tolerant rice lines. We got 12 genotypes with the highest percent of germination in the third quartile of the entire population. NHT25 is the best genotype. The new finding of this research is the moderate tolerant rice lines tend to be low temperature tolerant based on screening using thermogradient. Okay, thank you, and we are open to constructive input for the improvement of future research. We return the screen to the moderator.
Yes, thank you very much, Miss Penny Lestari. Next, we will move to the next presenter, that is Miss Hartati. She will present about the phylogenetic study of Gyrinopsis perstegi. So, Miss Hartati, screen is yours.
Assalamualaikum warahmatullahi wabarakatuh. Thank you to the moderator for giving us the opportunity to present our work. Hi to everyone. I would like to present our research on phylogenetic study of Gyrinopsis perstegi, the agarwood producing tree from Indonesia. This project is a part of my doctoral dissertation that supervised by Professor Iskandar, Professor Ulfah, Dr. Sri Hartati, and Dr. Samsida Rahmawati. Gyrinopsis perstegi is the endemic Thymilaceae species from Indonesia that known contribute to the global agarwood trade and become one of the most important agarwood producer. This species is distributed in eastern Indonesia, particularly in Maluku, Papua, Lesser Sunda, and Sulawesi. Due to the widespread illegal logging trees in their natural habitat, Gyrinopsis perstegi has been listed as the CITES endangered species. The major problem of Gyrinopsis perstegi are a rapid decline in the population of agarwood trees in their habitat, uncertainties in species identification required for conservation purposes, lack of genomic data needed to differentiate the species, and unresolved phylogenetic relationship between Gyrinopsis and other Aquarella species. Long-read sequencing technology such as MinION from Oxford Nanopore Technology has improved the ability to identify three genome species by directly sequencing a single DNA molecule in real time, allowing generating long reads. The advantage in applying this technology are producing sufficient overlap sequence from long-read DNA fragment, enabling the use of native DNA samples, portable and simple to prepare genomic library. Our research aim to conduct the phylogenetic or phylogenomic study on 6 Gyrinopsis perstegi collected from several areas of region Indonesia based on MinION long-read data regenerated with Oxford Nanopore Technology. We use 6 Gyrinopsis perstegi collected from several areas in Indonesia to conduct the phylogenetic study based on ONT long-read sequence dataset. High-quality DNA genome of Gyrinopsis perstegi was extracted from leaf using gene kit and CTAP method. The library preparation was conducted following the protocol developed by Nanopore for native barcode genomic DNA. Sequencing was carried out in flow cell Flomin 106 and then produced an output of long-read sequence in the FAST5 form. The signal in FAST5 form is first called to produce FASTQ data sequence. All reads were analyzed using the NanoGalaxy platform. The quality, statistic, and distribution of read before and after analysis were evaluation using the nanoplot program. The NOPO assembly was performed using the FLY. The resulting assembly statistics were calculated using QUAS and BASCO with Gyrinopsis walla chloroplast as a reference to check the genome fraction covered by the scaffold. The scaffold assembly were annotated using the GESEC platform for organellar genomes. Phylogenetic analysis was conducted using Eucalyptus grandis an outgroup, while Gonistilus affinis and Daphne tangutica were used as a sister group. The chloroplast genome sequence for aligned using MAF and bioedit sequence alignment editor. Evolutionary model were tested using the maximum likelihood statistical method to determine the most suitable model to construct phylogenetic tree with mega 7. The best fit for the chloroplast genome. The best fit model for the chloroplast genome was Tamura-Ney model, while for MATK and RBCL dataset, in the combination of both of them were Tamura 3 parameter. The bootstrap was determined using 1000 time replication. All data sequence from our study were deposited in the DNA database bank of Japan that linked to the NCBI. The longest contact from our study were used to construct the phylogenetic or phylogenomic study together with the sequence taken from the NCBI database. The phylogenetic tree was constructed using MATK or RBCL gene or the combination both of them, as well as the chloroplast genome from our study together with the sequence taken from the NCBI database. Based on the dendrogram constructed using MATK gene, it's shown that Eucalyptus grandis perform as an outgroup and separate all Timilaceae species into a different clade. All Timilaceae species were split into 2 distinct clades. Our results show that using the MATK sequence was insufficient to differentiate between Gyrinopstigii to most of the known Aquilaria or Gyrinop species from the NCBI database that used in this analysis. With only a 20% bootstrap support, Daphne tangutica was separated from the genera Aquilaria and Gyrinop. Gyrinop and Aquilaria species were overlapped into 2 clusters with the bootstrap support 16%. The low bootstrap support in the phylogenetic tree indicated that the phylogenetic relationship of Gyrinop and Aquilaria using MATK was not resolved properly. Although Gyrinop is always mentioned as a sister group of Aquilaria, in fact, the species of these 2 genera are very similar. Phylogenetic analysis based on MATK show that Gyrinop was paraphyletic to Aquilaria. Our finding based on RBCL gene show that the moderate bootstrap support in the phylogenetic tree indicated that the phylogenetic relationship of Gyrinop and Aquilaria using the single gene RBCL was still not resolved properly. With RBCL, Gyrinopstigii is LD2 and Daphne tangutica, which was placed outside of the Timilaceae group, its species in the clade of Timilaceae were also overlapping. Similar with MATK, RBCL is also proposed as a universal DNA barcode. Marker with good resolution should be able to discriminate between different taxa at both the species and genus level, and place taxa from the same species or genus in the same clade. However, the application of either the sole MATK or ABCL gene for Gyrinopstigii species demonstrate that it's insufficient resolution to identify one taxon to another for species identification. The utilization of both RBCL and MATK gene gave a similar result to the application of MATK alone. According to the phylogenetic tree constructed with both MATK and RBCL, Gyrinop and Aquilaria species were still overlapped in the same clade. Gyrinop and Aquilaria species were overlapped into 2 clusters with 55 bootstrap support. The position of Gyrinopstigii KRB 232 and 117A from our study were in the same cluster with Gyrinop caudata, Gyrinop koala, Aquilaria malacensis, and Gyrinopstigii taken from the NCBI database, while Gyrinopstigii LD2 was in the same cluster with the majority of Aquilaria species. Multiple locus genes have reportedly been recommended in to increase the resolution of genus distinction in Thymelaceae taxa using DNA coding. The phylogenetic relationship with low bootstrap support at the tree branch using both MATK and RBCL show that the distinction between Girona and Aquilera is still not resolved yet. Based on the chloroplast genome sequence, the sister and the outgroup of Aquilaria and Gironop were placed to separate clade. We found that most of Girinoperstigi were split into different subcluster from Aquilaria with bootstrap value 78%, with the exception of Girinoperstigi SLD2, which is in the same clade as Aquilera sinensis and Aquilera casna with bootstrap value 100%. The phylogenetic tree revealed that the species Girinop and Aquilaria were not totally separated since the Girinop cluster overlapped with Aquilera cluster. For species that are very similar and only distinguished by one character, such as the Girinop and Aquilera group, the grouping between taxon become more complex. Our results show that Girinop, including Girinoperstigia from our study, is paraphyletic to Aquilaria. The use of multilocus marker was proposed because it was considered more successful in identifying species, but in our sample, the combination of multilocus were still insufficient for other species. Then we used the whole genome chloroplast for species discrimination. Based on our finding, by using 6 draft genome of the chloroplast Girinoperstigi, we still found the same problem. The identification of Girinoperstigi has still been unresolved. From 6 draft genome of Girinoperstigi chloroplast that was used to construct the phylogenetic tree, 5 samples were clustered in one clade, while 1 sample was clustered in the other clade together with Girinopoala, Krasna, and Aquilera sinensis. In the future, we propose comparing the complete chloroplast genome of several Girinoperstigi to determine their taxonomic position in the Thymelaceae family. The entire chloroplast genome will provide a more accurate taxonomic position in the Thymelaceae family. Our result revealed that utilization of a single RBCL or MATK gene or a combination of both MATK and RBCL or a draft chloroplast genome are insufficient to distinguish Girinoperstigi from Aquilaria and other Girinop species. According to our phylogenetic study using stick draft
Okay, it seems the video is stopped. I think, thank you, Ms. Hartati. Yeah, we will go to the last but not least presenter, that is Ms. Nisa Yulianti. She will talk about the molecular docking. Ms. Nisa, screen is yours.
Hello, everyone. On this occasion, I would like to present our research on bioinformatics and molecular docking studies of germ-bound targets in breast cancer cells. These are our team. Breast cancer has been a threat for women across the world. Its prevalence is the highest among all other cancer types. It has affected almost 50 out of 100,000 persons per year in 2020. The molecular pathogenesis of breast cancers involves numbers of biochemical pathways as depicted in the figure. It also involves multiple signaling molecules and receptor. Interruption of one of those by using drug can result in overactivation of others that can cause drug resistance. Therefore, finding a drug that can targets multiple biochemical pathways can be a very good strategy to combating this disease. Zerumbon is a compound that is found in plants under Zingiber family, including Zingiber zerumbat, which is locally known as lempuyang. Lately, zerumbon has gained popularity among cancer researchers because it has been reported to have anti-cancer activity through several mechanisms, including induction of cell apoptosis and suppression of cancer metastasis and invasion through some receptor of signaling molecules. However, there is no research that comprehensively assesses as many targets that zerumbon can have in breast cancer cells. Therefore, our team aim to do research to find the potential molecular targets of zerumbon in silico. To do this, we first search for the possible target of zerumbon through stitch and string database, and we also search for the list of genes that involves in the regulation of breast cancer through NCBI database. And then we select for the proteins in common between those databases by using Venn diagram. After that, we assess for their protein-protein interaction with zerumbon with string, and then we scored that interaction by using Cytoscape and Cytohupa. And then we do the preparation for molecular docking by obtaining or downloading the structure of the target protein bound to small molecule inhibitor as the reference for the binding site through PDB database. And then we construct the 3D structure of zerumbon by using Avogadro, and then we optimize it by using Gaussian. And then we can do the molecular docking by using Autodoc, and then we assess for the binding type between zerumbon and the protein by using BioVia Discovery Studio. The result of our research is we got 83 protein that is the target of zerumbon and that involves in the regulation of breast cancer. And then we got the top 20 target based on the rank of their protein-protein interaction with zerumbon. However, we can only get the crystal structure of 6 protein out of 20 from PDB database. Therefore, we only do the molecular docking for these 6 with a different strategy for each of those. Firstly, for AKT1 protein kinase, this protein has been reported to statistically correlated with breast cancer disease status. For the molecular docking of AKT1 with zerumbon, we use 2 kinds of binding site, namely ATP binding site and allosteric binding sites. We predict that zerumbon bind to these 2 binding sites through alkyl p-alkene in ATP binding sites and through hydrogen bond in allosteric binding site. But we also found that there is unfavorable interaction between them. Secondly, apoptotic factor caps that code for caspase 3 and caspase 9 apoptotic factor. And then the inhibitor, its inhibitor CIAP. For these 2 genes, the strategy is by doing molecular docking of zerumbon to CIAP as the inhibitor of CASP at 2 kinds of binding site, namely BIR2, which is the binding site of caspase 3, and BIR3, which is the binding site to caspase 9. By molecular docking of zerumbon to CIAP, we predict that how zerumbon can induce apoptosis. We predict that Serum bond can bind CF at pair 2 and pair 3 through alkyl and hydrogen bond at pair 2 and hydrogen bond and alkyl also in pair 3, but there is unfavorable interaction. Next is HSP90AA. This is a survival protein that has been found to promote tumor invasiveness and malignancy. Serum bond is predicted to bind this protein through alkyl and p-alkyl interaction. Next is MDM2. This is a ubiquitin protein ligase that inhibits p53, a tumor suppressant protein. This protein is found overexpressed in breast cancer cell. Serum bond can bind this protein through alkyl and p-alkyl interaction. The last is EP300. This is a protein that involved in cell growth and division. Its knockdown has been reported to inhibit cancer. Serum bond is predicted to bind EP300 through alkyl and p-alkyl interaction. For the binding energy of serum bond to those targets is found relatively low. However, still higher than the reference small molecule inhibitor. Therefore, to conclude our research, serum bond might inhibit breast cancer progression through multiple targets, including AKT1, CYP2, CYP3, HSP90AA1, MDM2, and EP300. In the near future, we will do further research. Firstly, we will molecularly dock serum bond derivatives to those targets in order to get lower binding energy. And then next, we will choose the best derivative, and then we will synthesize the derivative, and then we will do in vitro anti-cancer activity assay. That's all for me. This research has been done because of the financial support by Institut Teknologi Sumatera. Thank you very much for your attention.
All right. Thank you, Ms. Nisa. I think the video is stopped. It was more than 10 minutes. Thank you for all the presenters. The topic are very interesting. And now I would like to open the question and answer section. If you have any question, please write down in the chat box or raise your hand with the icon below your Zoom. Okay, Ms. Lisa, please. You can open your mic, please.
Sorry, thank you for the chance, Ms. Bernadetta. I have a question for Ms. Hartati. I'm very interested with your research, especially about the method for the DNA sequencing with ONT. This is the first time I hear about the method ONT, Oxford something. I didn't remember. What principle? I'm wondering what is the principle of the ONT in doing the DNA sequencing? Is it using the polymerase change, the polymerase reaction like PCR, or is it different? I think that's all for me. Thank you.
All right. Is it clear for Ms. Hartati?
Yes.
Yeah, you can answer now.
In our case, we use non-PCR sequencing, but actually nanopore produce two type of kit, yeah, produce the two type of kit that can use for PCR or non-PCR base. So in this in our research, we use the directly sequencing using native DNA. So one of the advantages of using nanopore is the possibility to use native DNA directly to make a DNA library and then sequence with a nanopore. So it's quite simple and affordable, and also it's also can use on-site analysis. So this technique can use to sequence will be give a good advantage mostly for to sequence a tropical plant. Yeah. Thank you. Is it enough or?
I think it's enough. Thank you, Ms. Hartati.
Okay, thank you, Ms. Hartati, and thank you, Ms. Lisa, for the question. Is there any other question? All right, if not, maybe I can ask first for Ms. Yasanti. Yeah, and it's very interesting because the plant is endemic in the tropical country, and there are a lot of types of Nepenthes, I think, and then the status is from least concern until vulnerable. Okay, and I want to know what is the function of chlorophyll and carotenoid in Nepenthes, because maybe there is different function compared to other plants. Yes, Miss Yasanti?
Thank you for the question, Miss Benedita. Actually, the function of chlorophyll for Nepenthes is not too much different with another plant, but in Nepenthes, in leaf it differ to one leaf it contain one lamina and picture, and the color of picture is depend of insect which trap to the picture. And this study actually to find a better solvent to detect chlorophyll in lamina and picture. So actually we do two treatment, but only one in published because the one treatment in another publication. In picture, the color is different. One species can have a different picture. One species, yes, one species as the Nepenthes gracilis can make a color red or green dependent of the insect trapped in the picture. Nah, we want to know another factor besides that, so we do analysis chlorophyll. Besides that, analysis chlorophyll in Nepenthes also to know effect environmental situation in there. I think that's.
Okay, thank you very much. That's very interesting. Is there any other question? Okay, Miss Penny Lestari. Yes, please.
Yeah. Thank you, Ms. Bernadetta. I also interested with Ms. Yasanti presentation about Nepenthes. That as I know, to extract the chlorophyll from several species using acetone is the popular method. But in this presentation, Ms. Yasanti showed that the MSO is the better solvent for the chlorophyll analysis. Is it right? And
Yes.
it can be done for fresh and dry leaf. But the dry leaf that you mean is referred to silica gel, not open. So so my question is, while we go to the field and take some Nepenthes with very high water content, since it's Bali. Nah, how many silica gel, or how to treat the sample with silica gel until it can be extracted for the chlorophyll? Thank you.
Okay. Actually, I use around 200 gram for one leaf, around about, I forget for the luasan. When I go to Bangka Belitung for collect the sample, for collect one leaf, 1 until 2 leaf Nepenthes, I use around 200 grams leaf, and it can 1 week. So after that I change, but I change the silica gel every 3 days. It is enough.
Oh yeah, thank you.
All right, thank you, Ms. Penny, for the question, and Ms. Yasanti for the answer. Next, I see in the chat box there is a question from Mr. Elok Rifki Firdiana from BRIN. He will ask to Ms. Nisa Yulianti, and the question is, what are the compounds in serum bone which act in breast cancer treatment according to molecular docking? Because as far as I know, the analysis is conducted by interacting the specific compound or ligand with the protein target. Ms. Nisa, time is yours.
Okay, thank you very much for the question, Ms. Elok. So it's true that we do the molecular docking by interacting the specific ligand with the protein target, and serum bone here is the specific chemical compound itself that is being the ligand for the molecular docking. So we construct the structure, the chemical structure of serum bone, and then we use the 3D chemical structure of serum bone for the molecular docking as the ligand. So the serum bone itself is the ligand. Is that answering the question, Ms. Ella?
Oh, okay, thank you. I think I misunderstood. Understood. I think I thought it was the plant. It itself
Yes.
the chemical compound, and you construct it yourself.
Yes.
Oh.
Yes. So
Is it
serum
difficult
bone
is it difficult to construct and then produce it if it's effectively?
No, not at all, because not at all, because for constructing the chemical structure of serum bone, we just have to find the chemical name of serum bone, and then we use special application for that. So I just use application simply for constructing the structure.
Okay. Thank you, Mr. Manita.
You're welcome. My pleasure.
Okay, sorry, there's Miss Ella Rifki Vidiana. So thank you also for Miss Mia for the question. I think we still have 2 minutes left, and then there is there any more question? Just 1 minute. Okay, if not, maybe we can just close. Okay. Oh, okay, there is additional sentence from Ms. Yasanti that because the signal is not very good, she wants to add that for 2 until 3 leaves, especially this is for Miss Penny Lestaria, I use around 200 to 300 gram, and I change is 1 to 2 days when silica gel become pink. But just for information, based on another our research, after 1 week, chlorophyll content decrease and carotenoid increase. So this is very
Recording in progress.
Welcome back to the main room, ladies and gentlemen. The next agenda is plenary sessions for this afternoon. Therefore, I may invite Dr. Ira Handayani as the moderator of these sessions. But before that, allow me to read short profile of Dr. Ira. Dr. Ira Handayani is currently a researcher in Research Center for Applied Microbiology, National Research and Innovation Agency, the Republic of Indonesia, who are interested in molecular genetics, microbiology, and bioinformatics. She obtained her bachelor degree in chemistry at University of 11 Maret, and she continued her master degree in biochemistry at Bandung Institute of Technology, or ITB, and also her PhD in chemical engineering at Tübingen University, Germany. Dr. Ira, time is yours to lead the sessions.
Okay, thank you, Ms. Aulia. Are you hear my voice? Okay. Hello, are you hear my voice?
Yes, we can hear you
Okay.
clearly.
Okay.
Dr. Ira.
I don't know whether Yvonne already in here or not.
Yes, good afternoon, Ian.
Oh,
I hear you, and I'm already here.
Okay, how are you?
Thanks, fine. Hope you too.
Okay. Okay, thank you. Okay, I will start the presentation. The plenary session for this seminar. First of all, I'm very happy to see you here and welcome all of you to this plenary session. Before the presentation starts, let me tell you how the presentation will go. First, our first speaker, Professor Yvonne Mas, will be invited to present the material, and then our second speaker, Professor Claudia Tiribelli, will also be asked to present his material. After both speakers present their material, there will be a question and answer session followed by a conclusion. Now let's start with our first speaker, Professor Dr. Yvonne Mas. Okay, this is the CV of Professor Dr. Mas. Professor Dr. Mas is a professor of applied microbiology at the Technical University of Brunswick and head of the Department of Bioresources for Bioeconomy and Health Research at the Leibniz Institute, the SMZ, a German collection of macroorganisms and cell culture. She has experience as an editor in the journal Molecule and the journal Printer in Microbiology, and experiment as a member of the topical advisory panel in the Journal of Antibiotics. She is interested in research in the field of microbiology, biotechnology, actinomycetes, antibiotic, and regulation. Now allow me to welcome Professor Dr. Yvonne Mas to make her presentation. Professor Mas, please explain your material. And time is yours, Yvonne.
So thanks, Ira, for this nice introduction, and good afternoon to everybody. Okay, now I see my slides. Okay. Okay. So again, good afternoon to everybody. Yeah, thanks for the nice introduction. Actually, I'm here at the Leibniz Institute, DSMC, so the German collection of microorganisms and cell cultures in Braunschweig, Germany. And it's a great honor to give a talk during your session at the International Symposium of Innovative Bioproduction in Indonesia. And today I would like to introduce you into the topic of how we can exploit the biodiversity for novel natural compound production by using regulatory switches. So I should, now it's okay. So before I go into that more in detail, I first want to mention why this kind of research is relevant at all. And I think all of you know that antibiotic research indeed is relevant because we observe growing crisis from antibiotic-resistant bacteria. So the World Health Organization regards the antibiotic resistance disease as one of the 3 biggest threats to global health. So of course, it's urgently needed to find novel antibiotics to combat these drug resistances. But the question is where to find them. I'm sorry, the presentation is a bit slow, but I hope we can do that.
I think it's okay.
Okay. So we can, of course, find antibiotics a lot in actinomycetes. So actinomycetes are known to produce 2/3 of all antibiotic in clinical use today. And this antibiotic production potential is known for quite a long time. However, what is not known for such a long time is that the genetic potential for the production of antibiotics is even larger in these organisms. And this appeared when the first genome sequence of such an antibiotic producer has been sequenced. So this was the genome sequence of the model organism, Streptomyces silicolor. And that silicolor strain is known to produce up to 2 to 4 secondary metabolites under standard lab conditions. However, the genome sequence analysis revealed that the genome harbors up to 20 different biosynthetic gene clusters. However, not all of those gene clusters are expressed, meaning not all of these substances are really produced, but a lot of them occur as so-called silent antibiotic gene clusters. And this is not only true for that organism, but nowadays, since we have a lot of, more than hundreds of genome sequences available from different actinomycetes, it turned out that this is true for a lot of these producing organisms. So obviously, actinomycetes still hold a huge potential for the production of novel secondary metabolites. And of course, the current trend is to activate these kind of silent gene clusters. And now I would like to introduce you into the topic, how we can activate those kind of silent gene clusters in actinomycetes. Okay, and we actually can do that by using regulators. And regulators indeed are the set screw to unlock the biosynthetic potential of these natural compound producers. However, the problem is just that there are so many different transcriptional regulators abundant in actinomycetes, especially in streptomycetes, and these can be of different classes, and they can interact in quite complex cascades with each other. And depending on genome sizes, those can actually be more than thousands of different transcription regulators. So the question is, which is the most suitable and most potent type of regulator that is suitable to be used as a general activator of antibiotic biosynthesis. And today I will focus on these subtype regulators, since we think that these are good candidates to be used as general activators of antibiotic biosynthesis. Okay, presentation is still slow, but okay. So first of all, I will introduce you where we found this type of regulators. And we indeed identified them in the antibiotic producer Streptomyces spiralis, which is kind of our working horse. So this strain produces the pristinamycin substance, the streptogramin antibiotic pristinamycin. And pristinamycin is actually 2 different types of antibiotics, the peptide antibiotic pristinamycin 1 and the polyketide antibiotic pristinamycin 2. And the Streptomyces spiralis strain produces those 2 substances in a specific ratio. They interact synergistically with each other, and a semi-synthetic derivative of that substance is on the market. It's called Synergid, and it's used as an antibiotic of last resort against highly resistant pathogens. And what we did back then was that we characterized the antibiotic biosynthetic gene cluster. And back then, this was the largest antibiotic gene cluster that has been described so far. And the genes that you can see here in red, they are needed to, they encode for the peptide antibiotic pristinamycin 1, and the blue ones encode for the polyketide antibiotic pristinamycin 2. So when we had a closer look into this antibiotic gene cluster, then we identified a set of 7 different transcriptional regulatory genes, and they belong to different types of regulatory families. And actually, this is quite a large number, because usually these kind of antibiotic gene clusters have one or two such kind of regulatory genes. And we studied the function of all of those regulators in detail. However, today I will only focus on this red-labeled PAPR2 regulator here, which belongs to the subtype family of regulators. So when we studied the function of this regulator more in detail, and there I need the next slide. Okay. First of all, we inactivated the gene and studied the pristinamycin production, and there we observed that the inactivation of the PAPR2 gene leads to complete loss of pristinamycin production in comparison to the wild type, which still nicely produced pristinamycin 1 and pristinamycin 2. So therefore, we knew that PAPR2 is an essential activator of pristinamycin biosynthesis. Ira, can I ask, are you the one who is moving the slides forward, or can I do that?
No, I don't know.
Okay,
no,
because it's.
I think it's a bit left in there.
Yeah, it's really several.
I will ask Sandy.
But I will. It's okay. I will just move on. So PAPR2 is an essential activator of pristinamycin biosynthesis. This was what I wanted to say. And we studied the function of that regulator also more in detail, and figured out where the regulator really binds within the antibiotic gene cluster. So therefore, we purified the PAPR2 regulator and did electrophoretic mobility shift assays, and thereby we could show that PAPR2 really binds to the promoter regions in front of the pristinamycin 1 and pristinamycin 2 structural genes. And we were also able to define a specific consensus sequence for such a binding. And by RT-PCR experiments, which are shown here at the right-hand side, we also could show that there's no transcription of the pristinamycin 1 and pristinamycin 2 structural genes in the PAPR2 mutant in comparison to the wild-type organism. So therefore, we knew that PAPR2 is a direct activator and an essential activator of pristinamycin biosynthesis in Streptomyces pristinus pluralis. And the next slide, which I hope comes in a few seconds, shows you where the regulator is situated within the regulatory signaling cascade. Because it's here located at the very bottom, it directly activates the pristinamycin biosynthesis, and itself it is regulated by a set of other transcriptional regulators where I will not go in detail. However, it's quite complex signaling cascade governed by microbial hormones called gamma-butyronolactons. And what I wanted to mention here is that understanding these kind of regulatory signaling cascades and the regulation mechanisms of antibiotic biosynthesis in general can also help to activate silent gene clusters. And this we observed when we overexpressed the PAPR2 regulator in a foreign strain. And back then, this was Streptomyces lividans. And this lividans strain should come also in a second. This lividans strain usually does not produce any secondary metabolite under standard lab conditions. So this you can see here on the left-hand side, the lividans strain with the anti-vector control. However, when we overexpress the PAPR2 regulator, then the lividans strain produces an intensively red pigmented substance. And this red pigment indeed is andecyl protegiasin. And andecyl protegiasin is a known product from Streptomyces lividans. However, it is usually not produced in the lab, so it's encoded by a kind of a silent gene cluster. And this andecyl protegiasin biosynthesis is known to be under control of a subtype regulator called RED-D. And this RED-D shows quite high sequence homology to our PAPR2 regulator, so up to 55% sequence homology. And therefore, we speculated that PAPR2 takes over the function of RED-D and leads to the activation of this silent andecyl protegiasin biosynthesis in Streptomyces lividans. And we also later on could show that on the molecular level. So I'm really sorry that it's hanging, but yeah, this is what is shown in here. So having that in mind, we had a closer look into this kind of subtype regulator families in general. So in literature, they are described as streptomyces antibiotic regulatory proteins. They are known to act as pathway-specific activators of antibiotic biosynthesis. And all of them contain a conserved N-terminal DNA binding domain. And with this, they bind to conserved consensus sequences at the promoter DNAs of the promoter regions in front of the target DNAs. However, so far, the regulatory mechanisms, how those regulators really work, is totally unclear. So this is something what we are going to study in the near future. So we will do crystallizations of the regulator and try to figure out how this activation mechanism really works out in the antibiotic producers. So having that in mind, we had a closer look where those subtype regulators occur, and we found that the genes are very abundant in many different actinomycetes. So you can find them in nearly all streptomycetes, but also in other important antibiotic producers belonging to the actinobacteria family. And when you have a closer look where they occur within the genome, then you can find them as part of biosynthetic gene clusters. And actually, as part of gene clusters that encode for different types of secondary metabolites like beta-lactam antibiotics, phosphonate antibiotics, polyketides, and so on. So therefore, we knew, okay, the subgenes are abundant in very different actinomycetes. They are part of gene clusters encoding for different types of secondary metabolites, and therefore we said that those ones are good candidates to be used as general activators of antibiotic biosynthesis. And then we tested this out in a random strain in just a running project of ours, and this also will be shown, I hope, in a few seconds, because back then this was a project which we performed with some Indonesian partners. So actually several ones that are sitting in the audience today. So with Puspita Listianti, Vin Kusaryoto, Iran Dayan, Shantaratna Komala, and so on. So back then we aimed to isolate new antibiotics from Indonesian actinomycetes. So there we are. Back then, the Indonesian colleagues isolated up to 422 actinomycetes from Indonesian habitats. A subset of strains was handed over to our labs back then at the University of Tübingen, and we prioritized the strains for the most phylogenetically unique ones. We grew them in several different cultivation media and covered all of that to bioactivity tests to prioritize the one with the best bioactivity. And back then, 9 strains were of most interest. And from these ones, we isolated the genomic DNA of, did biosequencing, and then performed a combined genome mining molecular networking approach. And I think this was what Ira mentioned some hours ago in her talk. So this we have done for dereplication purposes and focus on the most unique gene clusters that encode for the potential unique substances. And back then, one of the interesting strains that turned out from this analysis was SHP-227, which showed quite some good activity. And we just randomly used that strain and performed our sub-guided activation strategy with that. So we cloned in our PAPR2 overexpression strain to SHP-227 and got extracts of the cultivation of these strains and tested it against Bacillus subtilis as a test organism, as you can see in here. The SHP-227 PAPR2 overexpression strain showed significantly improved bioactivity against Bacillus subtilis in comparison to the control strains with the anti-vector or the wild-type samples, which means that PAPR2 overexpression led to significantly improved bioactivity of that SHP-227 strain. Now, what you would do in the next step is that you go into the lab and do elaborative isolation of that compound and check what the substance really is. But we didn't do that. In the first place, we started a genome mining approach of that strain in order to find out what might be the substance that encodes for this, or what might be the cluster that encodes for the substance with this improved bioactivity. And as I said, we had the genome sequence in hand. We performed an anti-smash analysis for the prediction of potential antibiotic gene clusters, and thereby we identified 25 potential secondary metabolite gene clusters. And then the question was, which is the one that encodes for this bioactive substance? And then we ran an additional bioinformatic analysis by applying the program PET scan and searched for our PAPA2 consensus sequence. And thereby we found this subtype sequence in front of gene cluster 9. And gene cluster 9 encodes for potential nucleoside antibiotic, which has similarity to a gene cluster from a Streptomyces lineaceus strain. And that one is known to produce the substances amicetin and plicacetin, which are known compounds. They act as protein synthesis inhibitors with quite good bioactivities. And only when we knew that, then we went into the lab and checked by HPLC analysis what the product of this strain really is. This should be shown in the next slide, hopefully in a few seconds.
Sorry, Yvonne, I'm interrupted. Your time is 3 minutes again.
Okay, I will hurry up.
Sorry.
And I will go through the slides. Okay. So back then, we indeed could show that it's amicetin plicacetin that is produced by the PAPA2 overexpression strain and in significantly higher amounts. In the next slide, it is shown that this also helped for the isolation of 4 novel pyrimidine nucleoside antibiotics, which was a collaborative work with the group of Harald Groß at the Pharmaceutical Institute of the University of Tübingen. I'm really sorry, but the slides are just too slow. So this was just published in the beginning of this year. And now we know that this SARP activation strategy really works out. We are now having a set of different overexpression plasmids. I will skip that, but I hope, yeah, I hope that will work. So what we are just doing at the moment is that we apply this to the genome sequenced strains of the DSMC. So we scale that activation strategy up, and we already screened hundreds of genome sequenced DSMC strains for the abundance of SARP genes and SARP consensus sequences. And these are the ones that we are currently using for performing our SARP guided activation strategy. And in one of the next slides, hopefully, I still have the time that also, I will skip that one, that also in our selected DSMC strains, where we clone in our PAPA2 regulator, we observe significantly increased bioactivities in comparison to the control strains. So when we test different test strains as for the bioactivity. And for some candidate strains, we already have some potential substances that are produced. For several strains, you can already see by the eye that something is going on, that when you clone in the PAPA2 construct, that there's significantly production, or that there's production of a pigment in comparison to the wild type strain. And we are currently isolating these substances and trying to find out if there's a novel antibiotic that is produced by those strains. And with this, I'm at the end. I'm really sorry that the slides were kind of delayed, but I would like to thank all the people that have been involved in this project, mainly Ira Handayan, who worked a lot with these Indonesian strains, especially with the SHP27 strain. Also, Janina Krause performed a lot of these SARP overexpression analysis, and currently the right-hand person are involved in SARP-guided activation strategy of the DSMC strains. And with that, I would like to thank for your attention. I hope we can move on with joint collaborations because I really enjoyed that interaction with you. Okay, so thanks, and I would be happy to take questions later on.
Okay, thank you, Yvonne. Thank you very much for giving such an informative and exciting presentation. Now let me invite our second speaker, Professor Claudio Tiribili, MD, PhD.
Here I am.
Okay. Professor Tiribili, okay. Okay. Wait a minute. Okay. 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 Thank you. Professor Tiribili is the scientific director at the Fondazione Italiano Fegato. He has
Italian Liver Foundation. That's easier.
Oh, okay. Sorry. He has experience as an editor in the Journal of Hepatology and the Annals of Hepatology Journal, and experience as an associate editor in the BBC Research Note Journal. He is interested in research in bilirubin, neonatal jaundice, metabolic liver diseases, hepatocellular carcinoma, stem cell in hepatocellular carcinoma, Klicker nasal syndrome, and translation medicine. Now allow me to welcome our second speaker, Professor Claudia Tiribili, MD, PhD, to make his presentation. Professor Tiribili, time is yours to give a lecture now. Thank you.
Well, can you hear me?
Yes, of course. We can hear you.
Thank you very much for this kind introduction. Let me try to share the screen with you. That's already done. And so I can start my presentation. Let me tell you, it's really a pleasure to be a speaker of the 9th International Symposium on Innovative Bioproduction in Indonesia on Biotechnology and Bioengineering. What I'm trying to address today, I'm driving the presentation. What I'm trying to do today is actually make a link to what we are eating and drinking and the liver damage. As you know, no, it doesn't work. So why don't we remove my presentation? I'll share my screen.
Okay. Stanley, can you help me?
Yeah, I mean, I need to go on. Let me try to share my screen. And then, here we go. Okay.
So it's work now?
Yeah.
Okay.
Can you see it?
Yeah,
okay.
I'm seeing it now.
Okay. Let's move back to the start. Sorry for that, but okay. So liver is after the skin is the largest organ in our body, and in an adult weighs about 1.5 kilo. Although it's not that thick, the normal liver has a volume similar to the soccer ball. And during this period, we are very fan of a soccer ball because it's the largest gland in our body, and 80% is composed by the liver, by the hepatocytes. But most important, the liver is the organ in our body which is the largest number of functions. And liver can do more than 600 functions, such homeostatic, metabolic, detoxificant, and immunological function. But in addition to that, and not only a marginal issue, is that the liver is the only organ in our body able to reproduce and regenerate. And this is a Prometeo myth is being depicted. The main causes of chronic liver disease are different things, such as diet, here, medication, viruses, and autoimmune liver disease, moving from normal liver to usually fatty liver, which is increased presence of the fat in the liver, which can go on with the steatohepatitis and then go to liver cirrhosis, which is advanced liver fibrosis and functional complication, ending up with the strip with the presence of hepatocellular carcinoma, which is the end-stage liver disease. We need to stop this trip from here to here. And what I'm going to address today mainly is the role of eating and the diet in having liver disease. So And this is why, because MAFL, the so-called metabolic fatty liver disease, this associated metabolic liver disease, has a prevalence globally of more 25% of the population, which means in one out of the four European citizen has a fatty liver, so has a possibility to develop a serious liver disease. So if we go from the trip of from normal liver to hepatocellular carcinoma goes via different steps. First of all, we have the MAFL, the non-alcoholic liver, alcoholic fatty liver disease, which is, mainly represented by the increased amount of fat into the liver cell. But once the inflammation occurs, due most probably to oxidative stress, we have a sort of activation of the extracellular matrix of the liver, which is able to create scares and then move from NASH to cirrhosis, which is, labeled by the presence of scars in the liver, making the liver hard and less active. And these scars, together with the regeneration of the liver, end up in the hepatocellular carcinoma, which, as I told you, is the end stage of this trip. We have several risk factors for this. Of course, we have the overweight, the obesity. I'm going to touch this in more details later on. We have type 2 diabetes and insulin resistance. Altogether, overweight or obesity, type 2 diabetes, and insulin resistance make what we know now is called the metabolic syndrome. And the metabolic syndrome is related both in adults, but even more worrisome, is present also in children. And this as something we have to think about, because being in children, we need to prevent the fact that our child moves from here to here in the life span he or she is expected. And if we look here, that's very remarkable figure, that if you look the prevalence of obesity from 1975 to 2016, both in females and males, you may see the dramatic increase in the prevalence of obesity during this year, meaning that the more so-called civilized we are, the more fat we are going to be. And this is something we have to cope with and we have to fight with. And actually, even more worrisome is the trends in the number of children and adolescents with obesity. And if you both in males and in females, this is true all over the world. But if you calculate here, which is the Southeast Asia, also in the Southeast Asia, particularly in the male children are associated with an increased level of fat in the body. And if you look around the world, the presence of the metabolic fatty liver is basically almost the same all over the world. In some regions it's higher, like in US, in South America. In some regions it's lower, it's Africa, due to the fact there is a reduction in the calories available. Australia is basically like Europe, but in China we are observing now a booming wave of obesity, and this is something we have to cope with. So going back to what, in my opinion, is the most important aspect of the obesity is the so-called vicious circle of childhood obesity. We have a healthy child which actually due to a wrong life habits, sedentary life, lack of physical activity, is getting mildly obese child. And the mildly obese is actually a child is less prone to exercise, and those goes from mild to moderately obese child. And if this is also keep going with a wrong diet, is going to move from moderately obese child to severely obese child, and actually to and then to obese adult. And this is where we are going to go from a healthy child to a potentially sick child with the presence of fatty liver. And if you look around, you see that genetics may play a role in obesity. And we know now that the contribution of heredity to the risk of obesity is 25%. And this variability is related to the genetic background of the different studies performed. So we need to understand that habits, in addition to genetics, is the main predisposing factors, particularly habits of parents living with the child. And we just published an editorial in an international peer-reviewed journal, it was APT, making clear that it's the parents giving us the wrong information how to behave, less exercise and much more sedentary life. What are the targets of fat accumulation in the body? Of course, obesity is a multi-organ and multifactorial disorder. We can have damage in the liver, in the brain, in the heart, in the vessels, in the nervous system, and in the pancreas, and in the muscles. All together, meaning that we have a lot of complications of obesity in the childhood. We have damages in the behavior, psychosocial, neurological, like pseudotumor. We have pulmonary, sleep apnea, asthma, and intolerance of the exercise. Of course, cardiovascular, due to the dyslipidemia, hypertension, coagulopathy, gastrointestinal, as I told you about steatohepatitis, and gallstones, renal, endocrine, and muscle. So this, we have to conclude that an obese children, like an obese adult, are sick, and we need to cope with. So which are the main causes? Is the imbalance between the calorie taken and the calorie expended. I'm used to say that the weight is like our bank account. If you eat, if you gain a lot of money, you don't spend, you get rich. But if you eat a lot of calories, but you spend a lot, you get poor. So we need to understand that globally, there's a striking decrease in physical activity. 1 in 5 boys and 1 in 10 girls is not having a real and appropriate exercise. And even more worrisome is that the amount of fruit eaten by our children is drastically reduced as compared to the ideal amount. Only 30% of boys or roughly 40% of girls eat a fruit a day, in spite of the fact that fruit is readily available all over the world. And the major suspect is sugar. Sugar, the white sugar, it's really being associated with the presence not only of obesity, but also liver disease. And the question is how much is too much? And we need to understand this because we can give advertising. And here is what we have. And what we have here in the slide is coming from the World Health Organization, telling us the content of teaspoons for each beverage. And mind that the WHO recommends consuming no more than 12 teaspoons per day. If you drink a Coca-Cola, you drink it's like you eat 8.5 teaspoons of sugars. If you eat a yogurt drink, it's like 7. But if you need an energy drink, you have 10. And of course, telling you that what we are using to give our children, the so-called sodas, are then maybe very dangerous. And here's what we have in as comparison. Of course, the blank is the water. There's no sugar. But if you look at Sprite and Coke, which is widely used, we have that 33.7 and 35 grams of sugar per each can. And even worse are Fanta and Schweppes when we go from 33 to 43 and 44 gram of sugar. So telling us that, we are not surrounded by a friendly beverage consumption. What is the total fat? The total fat should not exceed 30% of the total energy intake. And we have different kinds of fat. We have trans fats, the dangerous one, due to the so-called junk food. We have the saturated fat, which is related to coconut, palm oil, and alimentary, polyunsaturated skin, meat, and fruit. And we have then the unsaturated fat, which are the real good fat, that should be much higher than 90% of the total fat in intake we have every day. And this is important to prevent additional damage. How the excess of fat?
Sorry, Professor Tribali, I may interrupt you because your time is only 4 minutes again.
My time is still 5 minutes.
Oh, 5 minutes. Okay, 5 minutes again.
I mean, according to my watch, it's still 5 minutes.
Okay.
So I'll be on time. Don't worry.
Yeah.
So how the excess of fat and sugar impact the liver? This is a cartoon. It's a very complicated cartoon. I've had no time, unfortunately, to go through. But the key issue is that insulin resistance and increased intake for the diet bring together the possibility to accumulate fat in our liver. And this is what we have to deal with. So do we have animal models to study the juvenile fatty liver disease? And the answer is yes. This is work we have done in our lab, driven by Sylvia and Natalia and Pablo. And what they did, they reproduced the fatty liver, a juvenile fatty liver, in a mouse, giving the mouse a lot of fat and a lot of calories. And what we have, what they have been able to reproduce is a fully scaled presence of fibrosis, meaning telling that this may be a good model to deal with in order to understand if there is any good treatment to work with this. In addition to that, we know that fatty liver is really linked to a leaky gut, because the gut is living alone with a lot of toxins moving from the gut to the liver. And this is what we have observed, that in large intestine, we have a leaky, the presence of a less tight junction as compared to controls. So in conclusion, we can say that excessive sugar and fat since young age induce obesity-related multifactorial events that lead to progressive multi-organ dysfunction, fibrosis, leaky gut, altered glucose homeostasis, which means diabetes. And this is, and we have to realize that the progression in children is much faster than adults. So we need to take care of this aspect for the future generations. And we know now that in the future 20 years, all the liver transplantation will be related to fatty children at the moment. So we need to take action on that. So let me skip all this because I have no time here. That try to deal with the future perspective. We heard that insulin resistant, type 2 diabetes, and obesity is the accounts for metabolic syndrome, which is a booming of obesity. And to date, we are still missing something, but we need to do something as a diagnosis, screening, and efficient treatment. And the clock is still ticking. So thank you for attention. I think I did my job in time, and I used all my 20 minutes.
Okay, thank you very much, Professor Tribali. Thank you for giving us such an informative and interesting presentation. And ladies and gentlemen, now are the time for the question and answer session. We have 20 minutes for Q&A session. If you have question for both speaker, please raise your hand and or you can write the question in the chat room. Okay, I think we have wait a minute. I need to see. I think Miss Atika, are you ready to give your question?
Thank you, Dr. Yeah, my question is to Dr. Yvonne. She's gone. No. Yeah. Okay. So,
Yvonne, Yvonne, are you still in here?
I'm still here. I'm here. Yeah. All right. Okay. So my question is a bit practical, no, but I never done the analysis that like what you did. So my question is like how long actually to get from your analysis of biosynthetic gene cluster until the analysis of the mutant, if I'm not mistaken. It's like you did like cloning for all this experiment, right? If I'm not mistaken. So ideally, how long until you get the results? Thank you.
Yeah, thanks for the question. That's actually hard to say because it's mainly dependent on the strains to manipulate. Actinomycetes are not so easy to genetically manipulate, and depending on which kind of organism you have in hands, this really depends how fast you are. But concerning the whole process, depending if you have the genome sequence, this is the first step. Let's say if you have the genome sequence, then you could go for the anti-SNP analysis, and this is quite fast because you just upload the sequence on the anti-SNP tool, and then you directly get the output after a few minutes. But this is, let's say, pure bioinformatics. It can give you an estimation what kind of gene clusters may be encoded, but you then need to really have a closer look into that and study that more in detail and also manually. And then, I mean, nowadays we have these overexpression plasmids in hands, they are cloned, then you just need to introduce them into the actinomycetes. And if everything works well, you are maybe successful after 1 or 2 weeks. If it's a strain that is harder to treat, then it can take several months, I would say. But this is what where I skipped a slide, because what I wanted to mention is that nowadays we have different genetic tools. So we have different plasmids, which can be used either for protoplast transformation, which is a process how you can get DNA into the strains, or you can use the plasmids for conjugation, where you use the opportunity that you have an E. coli strain that can deliver the plasmid to the actinomycetes.
Okay, Yvonne, thank you for the answer. And in the chat room, I have 3 questions for Professor Tribeli. First question is, it is true that some herb like curcuma is help in maintain our liver health? Thank you. This is from Mr. Elok. Miss Elok, I think. Are you ready to answer?
Yeah, the answer is maybe. I mean, there's no real confirmation that any drug is preventing liver disease. There are some drugs used to or suggested to be helpful, but I mean the real scientific demonstration is not yet. The curcuma is one. The other one is silymarine, the thistle syrup. Others are the extract from the apple. But I mean, there are some information, but not real specific and solid scientific demonstration. So we need to take this with a very, with caution.
Okay, thank you for the answer. Okay, and the second question is, what are the most recent and reliable treatment to deal with liver damage that are already on cirrhosis stage? Is surgery and transplantation are the only option?
Well, it depends about the stage of the disease. Of course, if you have an end-stage liver disease, transplantation may be an option. Actually, it is an option. We have to realize that in Europe, every year we are performing roughly 2,500 liver transplantation all over Europe. But I mean, the ideal is not to go to the liver transplantation. The ideal issue is to stop the trip before moving ahead. This is what I try to demonstrate in my presentation as far as obesity is concerned. If we start changing our lifestyles by asking our adolescents and ourselves to exercise more, to eat less, and to be much more pragmatic in terms of daily behavior, this could be very helpful. Not only for the liver, but also for the cardiovascular disease, for diabetes, for everything.
Okay, thank you for the answer. And next, are there any possibility to recover from any damage in the liver?
For what? For?
Are there any possibility to recover from any damage in the liver?
Absolutely. I mean, liver is a regenerating organ. If you have a acute hepatitis, I mean, and you heal from the hepatitis, I mean, like a hepatitis, the liver returns back totally to normal structure. So we don't have to have any scar. Of course, if you don't recover fully, for instance, from B hepatitis or C hepatitis or other diseases, then the situation is not acute, it's going to be chronic. So the damage is chronic. So the end result is that the liver is daily damaged by the heating stimulus. And this ends up with something which is chronic liver disease.
Okay, thank you for the answer. And now we still have 13 minutes left. If there are any question for both of speaker?
We have been too clear.
Okay.
Or too confused.
Okay.
I have no idea.
If there's no any question again, I will ask maybe to Professor. Okay, Professor Mas. I want to ask Professor Iwan, what medium was used for self-activation with genome sequence in DSMZ strand? Is there any effect of the medium on the successful use of the self strategy in activating the silent gene cluster?
Yeah, actually, also here we tested a different set of cultivation media just to make sure that we do not miss anything. So actually we tested 3 to 5 different cultivation media. Some of them you may know, I will not go in detail with these ones, but it turns out that if you have activation, that 1 or 2 cultivation media are enough. So it looks like that really the abundance of the subtype activator is more relevant than the media you use.
Oh, I see. Okay,
thank you, Yvonne. But Ira, I think I also see a question in the discussion panel or in the chat room
I will take
from Hatati. I may also read it by myself because the question was what kind of data that one inputs to get biosynthetic gene cluster using plantis mesh. So I'm not sure if this is if I understand the question correctly, because there are different tools available. So anti-mesh is the program that can predict antibiotic biosynthetic gene clusters encoded in bacteria, and plantis mesh is a tool where you can identify gene clusters from plants. So if this question really targets plantis mesh, and the question is which kind of biosynthetic gene cluster one can find there, I would say that you get an output which mainly contains gene clusters encoding for terpene substances, type 3 polykey-type gene clusters, alkaloids, and so on, because these are more substances or gene clusters that are more abundant in plants. In bacteria, you may get just another output also on gene clusters encoding for non-ribosomal peptides, type 1, type 2 polyketones, for example. So I hope I could answer that question.
Okay. Yeah, thank you, Yvonne. If there are any question again for the both speaker, please raise your hand or write it in chat box. Maybe I will ask Professor Claudia Terribili. Does consuming too much fiber also causes liver damage?
Yes. I mean, the point is not what you consume. The point is what the amount you consume and the calories. I mean, if you eat, let's say, a kilo of bread, the amount of calories is much higher than a kilo of salad. But of course, the taste of 1 kilo of bread is much better than the taste of 1 kilo of salad. And if you put some butter on your bread, the calories increase. So I think we need to make a balance of our daily behavior. I mean, if we eat more calories, we need to spend more calories. As I told you during my presentation, our weight and our metabolic functions is like the bank account. I mean, if you gain a little amount of money, but you are so able not to spend, you're getting rich. But if you gain a lot of money, but you spend a lot, you spend more, you get poor. So everything is balanced. We need to be careful. And then the basic point is to teach our children to behave properly. And our children, at least in Europe, don't behave properly. They are in front of a screen. They don't move. They don't go to walk. They don't go to play football. They don't go on the bike. They stay home, and this is bad. That's why we're having a real problem with it with adolescents.
Okay. My next question is there any effect of a supplement on the liver health?
You mean the supplements?
Yeah, like vitamin?
Well, I mean, the supplements have never been demonstrated to be helpful. The supplements are very helpful for the companies that are making the supplements. They're making a lot of money they're making a lot of good for the producers of the supplements, but there is no evidence that are making good for the liver of the subjects taking the supplement.
Okay, thank you.
So take a message, don't take supplements.
Oh, okay, thank you. Thank you for your suggestion.
Eat a fruit, it's more than enough.
Oh, I see.
You're lucky enough to live in a country like Indonesia, where the fruits is so beautiful. And you have mangoes, you have everything.
Yeah.
Why don't you have to take supplements? If you're living in Europe, I mean, you go and then eat an apple or an orange or a pear, and that's it. It's better, it's cheaper, it's much more healthy. Trust me.
Yes, of course. Thank you for your suggestion. Okay, if our time is left 2 minutes. Okay, or Yvonne, do you want to ask the question to Professor Tribble?
Yeah, actually, I would also want to ask a short question to Mr. Tiribelli. I wondered if there are any numbers. What is in general more harmful if one drinks too much alcohol, or using too much calories from early days on?
I didn't touch alcohol for obvious reasons because but alcohol, there is a safe amount of alcohol daily, which around 30 grams per day, both in females and males. 30 grams of alcohol per day means half a liter of wine or a small glass of super alcoholic schnapps or whatever it is, which means that if you have one glass of wine with your meal, it's good. What is bad is to drink outside the meal because the absorption of the alcohol without any aliment is very quick. This is why the heavy drinkers are used to eat together with alcohol something which is oily, like olives and peanuts, because you have a sort of film of oil in your stomach, so the absorption is less, and then you can drink more. That's the trick they're doing. So, a limited amount of alcohol is safe, actually. But, it depends on how limited is limited. And, a good glass of wine is good. And let me tell you that, unfortunately, I'm a teetotal. I cannot drink alcohol, not because I don't want, because I cannot stand it. And this is my limitation, because, with a good meal, a glass of wine, assuming that your religion is allowing to do it, it's good.
Okay, thank you for the answer. There's 3 questions again in the chat room. First, for Professor Tribble. The question is, as we are getting older, is it true that our metabolic rate are also slower than when we are young?
Yes. Yes, the answer is yes.
Yes, okay.
That's it.
Yeah. And there's 2 questions for Yvonne. Did you also conduct any research in Germany on food culture to activate gene cluster for the production of natural products? based on your experience, which substances from terrestrial and marine ecosystem produce more new metabolites at this time?
Okay, concerning question 1, we didn't do co-cultivation approaches, and I tell you why. Because later on, the identification of the produced metabolites is just too hard. If you do co-cultivation, you may activate something, but you do not know what is produced. And then you need to go again into the lab, isolate the substance, do chemical analysis to really identify the produced substance. We had the attempt to do a targeted activation, because if we use these kind of regulators, then we exactly know where they bind, which kind of antibiotic gene cluster is activated, and this facilitates later on the identification of the compound. And therefore, we didn't do co-cultivation, which also may work, but we only focus on this targeted activation approach. So the second question is, which kind of organisms do produce more antibiotics, terrestrial actinomycetes or the marine-derived ones? We cannot say until now. You can have marine organisms with really a lot of antibiotic gene clusters, but also terrestrial ones can harbor more than 50. So I don't see a difference in terms of the potency of terrestrial against marine organisms. So there's still enough out there to be identified in terms of new secondary metabolites, I would say. Okay, but I think there's an additional question. Ira, would you like to?
Okay, I think there is no any question again. Okay, I think time is finished. Time is the end for the question. Finally, we come to the end of the seminar. I want to draw a conclusion from what speaker said. From Professor Yvonne Mas, we can learn that a self-guide strategy can be used to activate antibiotic biosynthetic gene cluster in a targeted manner and thereby exploit the biosynthetic potential of actinomycetes. And from Professor Tribuli lecture, we can conclude that excessive sugar and fat consumption from a young age can lead to the obesity, leading to the development of metabolic associated fatty liver diseases and non-alcoholic fatty liver diseases in the children. Unfortunately, the development of metabolic associated fatty liver diseases in the children is faster than in adults, and efficient therapy to treat this disease doesn't yet exist. Hence, the best options still depend on the lifestyle changes. Okay, finally, I would like to thank to Professor Yvonne Mas and also Professor Claudia Tribuli for their informative and interesting presentation, and all the participants for their very active participation. And now I will hand over the event to the MC. Thank you. Miss Aulia, time is yours now. Thank you.
Terima kasih.
Terima kasih banyak.
Thank you, Professor Tribuli, Professor Yvonne Mas, and also Dr. Ira for the amazing and also fresh sessions this afternoon. Distinguished guests, ladies and gentlemen, now we have arrived to the end of today's conference. On behalf of official committee, I would like to thank you for your attendance and also active participations on the first day of ISIBio 2022. We're signing off. See you on the second day of 9th ISIBio tomorrow at 9 AM Jakarta time with another interesting speakers and also materials. Stay safe and also healthy.
Bye-bye. Thank you.
Thank you very much, Yvonne and Professor Tribuli.
Thank you.
Bye.
Bye-bye.
Bye-bye.